Okay, thank you very much for that introduction, David, and thank you very much to the organizers for inviting me to speak here today. It's a great pleasure to be here. I love this conference. I've been a couple of times before, and it's really exciting to be here in Berlin and have the conference in Berlin this year. So I'm really excited to be here and to tell you about what's happening in my lab. So I hope that the work I'm going to tell you about today is relevant to cognition very broadly, although as many of you know, in my lab we focus on decision making. And we mostly study decisions that lead to actions or to movements, and I think many of you in the audience would agree that that's a reasonable thing to do. And that's for a couple of reasons. So first of all, many decisions, probably most of the decisions that we make, just naturally give rise to action. If you see the mouse up in this image here, if he decides that he wants to eat the blueberries, that will naturally lead him to rear up so that he can acquire them, get his head closer to the fruit. The second reason that we study decisions in the context of movements is that it's really convenient from the point of view of experimental design, right? If you have a subject reporting their choice with a movement, you can track those movements, whether it's pressing a keyboard or making a saccade, and you can study their associated metrics, like how long the decision took, was it correct or incorrect, and so on. However, this approach of studying decisions in the context of movements has led to a known challenge, and that challenge is untangling the neural activity for movements from the neural activity that's related to other cognitive processes. And probably all of you are familiar with this challenge or maybe even grappled with it yourselves. There's lots and lots of instances. For instance, you might have monkeys making decisions about random dots and track very carefully the metrics of their movements and try to account for those in the neural data. You might be studying a rodent and track its head movements and see whether those are related to the choice that the animal makes. And the work that's been done so far is definitely important. It's been a really key aspect of studies of decision making, is that they're really thoughtful about these instructed movements that the animals or humans were trained to make and thinking about the impact of those instructed movements on neural activity. But what I hope to tell you today is that even though that approach is important, it doesn't go anywhere near far enough in thinking about the kinds of movements that are occurring during decision making and thinking about what they mean for neural activity. So let me just formalize this a little bit for you here. So suppose you're studying decision making in a mouse, and maybe you've trained the mouse to make a licking movement, say to the left or the right, to report one decision or another, lick to one spout or other. We would refer to this collection of movements, left versus right, as an instructed movement, because we trained the animal to make those movements. He's instructed to make them, and he does them to acquire rewards. Other instructed movements might be a handle grab, which is what we use in the task I'll tell you about today, that the animal uses to initiate a movement. However, there might, of course, be other kinds of movements, like uninstructed movements, that the animal was never trained to make. And in the example that I've highlighted here, the animal is making a hind limb movement, a couple of hind limb flexions, every time the visual stimulus comes on, and that's meant to be schematized by that little gray box that you can see right there. And so we started to wonder, well, do these uninstructed movements matter very much for interpreting neural activity? I think many of us sort of assume that they don't, partly because it's quite difficult to track uninstructed movements. It's already difficult enough tracking instructed movements. What about having a whole other class is going to make things rather difficult. But we wanted to know whether these movements mattered for neural activity. And once we started thinking about it, we realized that there were really two kinds of uninstructed movements: the task-aligned ones that I just described a moment ago, the ones that occur at the same time on every trial, like in response to the visual stimulus, kicking a leg, for instance. But there might also be task-independent uninstructed movements. That is, they occur idiosyncratically on each trial and don't really have anything to do with the structure of the trial. And so we wondered, are these uninstructed movements, are they mostly aligned to task or trial events, or are they mostly independent? And just to complete the matrix for you here, of course, the instructed movements that we taught the animal, those have a small task-independent component as well, because of course, even though we train the animal, make your decision when the beep sounds, there's a little bit of reaction time, so it varies a little bit from trial to trial. So the question that we set out to answer with this set of experiments that I'll tell you about is, do brains prioritize cognition and instructed movements, or do other signals dominate? So to do this, we developed an auditory visual detection task in mice. We've been studying decision-making in rodents in my lab for a number of years. We have a pretty large, extensive set of experiments that we've conducted, seeing how rodents and humans measure up, understanding what they base their decisions on, and so forth. In this particular experiment, we have a head-fixed animal that has to report the spatial location of an auditory or visual stimulus. It'll be on the right or the left. It'll be presented twice, and the animal reports its decision about where that stimulus is by making a licking movement to the left or the right. It's a fairly simple task, although there's a delay period, so the animal has to hold its decision in mind for a little while. And the stimulus can be either of two modalities. We're really interested in decision-making behavior apart from neural activity today. In work I won't have time to tell you about today, we've been studying decision lapses, why animals sometimes make mistakes on easy trials. So if you're interested in knowing more about behavior, please ask me in the discussion. Okay, so I'm going to show you a couple of videos now of the animal doing the task. So this is now a camera that's pointing at the mouse from underneath. So you can see its nose and mouth at the top of the screen there, and its paws are at the bottom. And in this video, you're going to see the animal initiate the trial by grabbing two little handles that move in by a servo motor. So there he goes. He makes contact to initiate the trial. And then this is now a view of the mouse from the opposite point of view. Now you're looking from the back, and that's his tail down at the bottom, and you're going to see a visual stimulus on the right. So he sees the visual stimulus presented twice, then there's a delay, he hangs out for a bit. Now we're back to the original view, and you're going to see the two little spouts move in. He's going to commit to one of them, and then the other one will move out of the way. It's going to happen really fast. So he commits to that choice, and we move the other spout out of the way with the servo motor just so that we prevent him from changing his mind. Once he makes a decision, he has to commit. Okay, so we hadn't really used video a lot for tracking behavior before in my lab, and this turned out to be really critical, as you'll see throughout the talk. Using these videos, we monitored a whole bunch of aspects of the animal's behavior: eye position, whisker position, other facial movements. You can get a sense of what that looks like here from the two cameras that we measure the behavior. This is the pupil diameter, which you can see is kind of fluctuating over time, even throughout the trial. And I should say that in using video making videos and using it as a tool to connect to neural activity, we were very inspired by work from Carson Stringer and Marius Pachitariu, who originally presented this at Cosine, where we learned about it, and now have a paper that came out earlier this year. Okay, so one of our goals that I just described a moment ago is that we want to understand how brains prioritize cognition and instructed movements, or if other signals dominate. And so to get at this, we felt it was important to look at cortical areas really broadly. We really wanted to look across the entire dorsal cortex and not restrict ourselves to a particular, to one or a small number of areas, which is sort of a more traditional approach in a lot of animal decision-making work. So what we did is we took transgenic animals that express a genetically encoded calcium indicator in all of their excitatory cortical neurons. So the upshot of this genetic label is that if we have a neuron, once it starts to fire, it goes from being not green to being green, and then we can capture the photons from that fluorescence with a widefield macroscope that we built. So this is a way of imaging neural activity in the brain that allows us to see activity all across the dorsal cortex. We capture the photons that come up just with a CMOS camera. And for aficionados in the audience, this is a two-channel setup. We have the green light the blue light that we use to activate the GCamp, collect the photons from that, and on alternating frames, we also use violet light, and that allows us to make a hemodynamic correction. We can take the difference between the two signals. It's fairly spatially precise. It's about 20 microns per pixel, which is pretty good spatial resolution, and it's about 30 hertz. So we have a 60 hertz camera, and we do alternating frames with the blue light and the violet light. So one really big win of this approach is that because the mouse has a fairly thin skull, we're able to image these calcium signals all across the brain through the skull. So we don't have to put in any kind of a window. It's a totally non-invasive procedure. We have a couple of tricks that we use to make the skull a little bit clearer. We borrowed those or adapted some techniques that were originally developed in Carl Svoboda's lab a few years ago. And you can see we get a really beautiful view of the vasculature without having to actually go into the brain, which is super convenient. So what we do, we take a mouse with the GCamp and the neurons, clear the skull, and the first thing we do is we want to make sure that these measurements are really giving us something reasonable. And that's pretty important because wide field imaging is a fairly new way of measuring neural activity. You want to have kind of some ground truth. And there, we're going to take advantage of the fact that in the mouse brain, space is represented multiple times, well, as it is in humans and primates as well. There are topographically organized maps of visual space, and there are lots and lots of them. A big win of the mouse is that in addition to having a very thin skull, it has a lissencephalic cortex, which means it's flat. There's no bumps or grooves. So we can go in with some nice horizontal bars, which we move vertically, and vertical bars, which we move horizontally. And then we look all the way across the dorsal cortex, which phase of the movement of the bar do the different pixels respond to. And pixels, of course, reflect the summed neural activity of many neurons. And this allows us to identify primary visual cortex, which you see here, and usually between 4 and 7 additional surrounding areas. We can then use the skull landmarks, because remember, it's an intact skull, to line up these responses to the Allen Institute Common Coordinate Frame, and this gives us further reassurance that we're seeing exactly the right signals in the right place at the right time. So we do this for all our mice. These are the mice that are going to be, most of the mice that are included in the study that I'm going to tell you about today, and we get pretty nice retinotopic maps in all of those mice, telling us that we're getting a good signal from the brain. So once we've confirmed that we have a good signal, then it's time to measure activity during the decision-making task that I mentioned to you a moment ago. So I'm going to show you a movie of what that looks like, and the white text at the bottom is going to tell you which period in the task you're seeing the data from. I've slowed down time quite a bit so that you can see the neural responses evolving. I should say before I showed you the movie that what we expected was that more than one area would be involved. After all, there's auditory signals, visual signals. These are all going to be from visual trials, but still, maybe a few areas would be involved. But we thought it would be fairly restricted, because after all, this is not a terribly difficult task. It's just a spatial localization task. But in fact, what we saw did not meet our expectations at all. So you can see even in the baseline activity, baseline period, we're getting a lot of fluorescence activity. Kind of cools off for a while. Stimulus comes on, that's primary visual cortex in the back of the brain that's lighting up. Delay period. And then the whole brain gets really, really active along the time of the decision. So this really wasn't what we expected to see. So we needed to kind of get to the bottom of what was really happening with this signal. So we did a bunch of processing on the data. We take the 200 highest variance dimensions, which we just compute with SVD. And this is just one more view of the raw fluorescent signal, which we collect. So the first thing we did is said, okay, let's just look at the trial averaged activity. Maybe we're just measuring some kind of crazy signal here. Let's look in V1, see what's happening, and see whether we're encountering some odd signals here. So we started off looking in V1, that's back here, and I'm just going to show you the raw fluorescent signal, that's what we call delta F over F, as a function of time, and I'm going to show you data from trials where the animal saw a visual stimulus and trials where the animal experienced a tone. So you should have active V1 for the visual, hopefully not for the tone. So this is at the beginning of the trial before the stimulus has come on. There's a red and a black trace there, but you can't see them because they're totally overlaid. And then as soon as the stimulus comes on, you can see there's a big difference between light-on trials and auditory trials. Really reassuring. So we're seeing exactly what we should in the trial averaged activity in primary visual cortex. At the end of the trial, that difference mostly goes away. And then we compared activity in left V1 and right V1 when there was a stimulus on the left. And again, you can see we get just the signals that we'd expect to see, a larger response when the V1 is, when the stimulus is in the contralateral hemifield. So this kind of gave us reassurance that we were measuring reasonable signals. So then we thought, well, let's get to the bottom of what's driving this enormous brain-wide activity that you all saw in the movie a moment ago. So we decided to build an encoding model, and our goal here was to take the activity of each pixel, like this one here, for instance, and if we knew what its activity was, say, on trial 1, trial 2, and trial 3, we wanted to build a model that would allow us to predict the pattern of activity on trials that were left out of fitting the model. So we wanted to understand all of those little bumps and wiggles that you see up there. I wasn't super optimistic because I've been recording neural activity for a long time. There's always lots of bumps and wiggles. We never really seem to get to the bottom of most of them. But we felt kind of encouraged because we have a lot of behavioral variables at our disposal. So we have all the traditional decision-making variables, the instructed movements like the handle grab and the licks, and of course the stimulus. And these vary a little bit from trial to trial, making it feasible to fit an encoding model. And we also had at our disposal the video data that we collected. So we, just like the widefield data, we reduced the video data, and we can, this is the top 3 PCs for 3 different trials. And that's another way another kind of handle that we had, opportunity that we had to understand what the neural activity was doing by using the video. So we just built an encoding model where all of these different kinds of behavioral parameters were regressors. We put them into a big design matrix and then fit the model using ridge regression to try to find a set of beta weights that would allow us to map the behavior to the neural activity and then to test that on left-out trials to see if we could predict activity on trials that we never trained the model on. So before I tell you how well the model did, I just want to draw your attention to the fact that there are two rather different classes of variables in this model. So the first type we call temporal event kernels, and the other kind we call filterless analog predictors. So the temporal event kernels are super flexible here, and they allow us to capture quite complex time-varying responses. These are really useful if you have events that are occurring at discrete moments in time, like the onset of the visual stimulus, a lick, and so on. And these are probably familiar to many of you. They've been used fairly successfully in electrophysiology and imaging data previously from the mouse, and are also quite popular in fMRI. The other kind of predictor are the filterless analog regressors, and these are for behavioral parameters that vary continuously over time. So for instance, pupil diameter isn't really a specific event for which you could make a kernel, it's just continuously varying. And these analog regressors will be really good if, for example, the relationship between the behavioral parameter and the neural signal is that one is like a scaled version of the other. If that's the situation, the analog regressors are going to totally crush it. But if there's any kind of temporal delay, they're going to fail. If there's any more complex dynamics, these filterless analog regressors will be pretty much useless. But we felt like we needed to use them because we had a lot of continuously varying parameters. Certainly the video is something that varies continuously, so the video is in that class of filterless analog regressors. Of course, we orthogonalized the video data to all of the other regressors so that we wouldn't be getting overlap in terms of which predictors were accounting for the neural variance. Okay, so back to the model. So now you know what the classes of variables are. Now I'm going to show you over at the right-hand side of the slide how well we were able to predict neural activity on left-out trials. That's what you can see in red. And surprisingly, the model worked pretty well. We were pretty encouraged to see this. These are just a few example trials, but this heat map shows you the cross-validated R-squared all across the brain. This is collapsed over time. Now I'll just unfold time a little bit. This is how well we did during the stimulus period, during the delay period, and so on. And you can see that there are certain parts of the brain where we captured the neural activity better than others in somewhat sensible ways, right? Like when the stimulus is on, we capture activity in V1 pretty well and so on. But overall, we were able to capture activity pretty well across time and pretty well across space. So this was quite encouraging. And so we said, okay, great, well, let's look under the hood of the model. Let's see like what was really working to make the model fit so well. So what we did is looked at the behavioral parameters one at a time and looked at the contribution of each behavioral parameter to our ability to predict the trial-to-trial fluctuations. So in this group here, we call these the task variables. They're sort of traditional decision-making variables, like when the stimulus came on, the animal's choice, and so on. These are the instructed movements, the class of movements people have considered previously that we trained the animal to make. These turned out to capture a fair bit of variance, so it's a good thing. People were thinking about them for so many years. But the surprise was, when we looked at the uninstructed movements, they were by far the most important. They totally crushed the other variables, outpacing them really at every moment in time all across the brain. There are a bunch of different movements that I've included in this category. They're rather different classes of movements, so we can talk in the discussion about whether they should all be lumped together or not. But I think what's clear is that these uninstructed movements that, as far as we know, have nothing to do with the task at all, we didn't tell the animal to do them, these are really dominating the neural activity. Particularly the video signal and the first derivative of the video, which is video motion energy at the far right. These were the most important variables for predicting what the neural activity was going to do. At this point, though, you might be skeptical because you might be worried that a lot of these behavioral parameters are related to each other. And so now it's time for Skeptic's Corner. Okay, so Skeptic's Corner, we're going to go back to the model and say, yes, it's true, the behavior parameters may be related to each other. We need a much more conservative approach. So what we're going to do, we're going to go through the behavioral parameters one by one, kick them out of the model, and we're going to say how much worse does the model get? Or another way of saying this is sort of how much does the model suffer, and where and when does the model suffer when we remove a particular variable? And this is a much more conservative approach. So just to give you an idea, when we fit a single parameter like the onset of the visual stimulus, you can see that it's not hugely localized to area V1, but then when we use this much more conservative approach, you can see that when we remove the visual stimulus onset, suddenly the model is much worse at predicting neural activity only in primary visual cortex. So this is kind of a useful way of cleaning up the signal and also gives us a much more conservative estimate of the contribution of each of these different parameters. Here's another example, the handle grab. You can see if you just look at the overall contribution of a rightwards handle grab, you see totally bilateral activity, but the reason for that is just because right handle grab and left handle grab tend to happen at the same time. So you see a much cleaner signal if you ask how much worse does the model get when you eliminate that variable. And finally, with the nose, again, the activity looks really distributed across the brain, but that's because nose movements are correlated with other movements, and when you look at the nose by itself, it's much more restricted. So we did this for all of the different behavioral parameters that we had. I'll bring this up again in the discussion if people want to stare at it. You can look for your favorite movement effector. This was really the most computationally intensive part of this study, and actually was the reason why I think it would have been rather difficult to have done this until recent days when we had a pretty fast HPCC at Cold Spring Harbor, because we had to, one at a time, eliminate the model, refit the model without that one, and do that many, many times so we could cross-validate. But we did it. And this meant we could then go back to the under the hood figure that I showed you a moment ago and say, well, now let's look at all these variables and see how much worse each of them gets when we remove them from the model. And that's these dark green bars here that are now downgoing. And what I hope is apparent is that for all of these task variables, it almost didn't matter at all. We barely needed them to fit the single trial neural activity. We could get rid of almost all of them and not see much of a detriment in model performance at all. Not entirely, right? You do need to know when the stimulus came on to understand V1, but the overall effect is really minimal compared to instructed movements or uninstructed movements. So if we look at the pooled contribution of each of these three groups of variables, you can see that the uninstructed movements are really outpacing all of the others. This plot is collapsed across space and time. I'm now going to unfold this over space, and I hope it's clear that all across the brain, the uninstructed movements are most important. In fact, if you look at V1, reassuringly, you need V1 to explain some of the task-related parameters, which makes perfect sense, but you capture really a lot more of the variance in V1 with uninstructed movements than you do with the actual stimulus parameters. One possibility is that these uninstructed movements are there to sort of support the instructed movements, but we can get at that by looking at their contribution over the course of time in the trial. You can see that uninstructed movements really always dominate, even very early in the trial before the stimulus has come on. Okay, so I've highlighted these uninstructed movements here, and they're clearly really important, but you might be wondering, well, what are they? And some of them we know, because some of them we captured with sensors, like hind limb flexions and pupil diameter and so on. But a lot of, as you recall, a lot of the variance was really captured by the video parameter. And so I want to point out here that our approach differs in a few ways from previous approaches. First, in the scale of the data we collected, and also in the number of behavioral parameters and movements that we considered. But also, it differs in another way too, which is that we took a really agnostic approach, right? We just said, well, we don't know what might matter in the video. We're just going to take all the video and in a totally unsupervised way, just see if it matters. And this was really useful because it turned out there were aspects of the video that we didn't know were important and really turned out to be. The downside of this is that we're left really without a convenient low-dimensional summary of what it was in the video that actually mattered. And this can be kind of frustrating. If you have a hypothesis about a particular effector that might matter, of course, you can track it with LEAP or Deep Lab Cut, or you can use methods like MoSeq to define poses that might matter. But this is a very kind of hypothesis-free way of looking at the data. And we do have some way to get a little bit of insight into what these movements were that mattered so much. We can relate the wide field data to the behavioral videos by looking at the weights that mapped one to the other. And then we can take these low-dimensional weights and project them back up into the full high-dimensional video, and then we can color code those with a heat map in terms of how high those weights are. So what do we learn from this? Well, I think a few things. First, there's a lot of pixels around the kind of jaw and eye that seem to matter. It seems that mice are making a lot of small facial movements that might even be present in subjects who, were you able to give them verbal instructions, were told to remain still. They're fairly small muscle groups that are making those movements. And one could, of course, get to the bottom of this. If you really are a movement person, you care about what those movements are, you could track those with much more deeply and with much more care. But on the other hand, you might be someone who's really interested in cognitive signals, whether it's decision-making or attention or whatever, and you might feel that a better strategy is just to take this video in an unsupervised way, use it to understand how movements are driving the data, and then correct the data accordingly. And I'll tell you a little bit more about how to do that in a moment. I should add that we're thinking really deeply about video and how it might be used to interpret neural activity. We have a collaboration with the Paninsky Lab that was recently accepted as a NeurIPS paper, using some really clever and sophisticated ways that they came up with to process the video data and relate it to neural activity. So I think what I've told you about today is really only just scratching the surface of a whole different way of looking at behavior. Okay, so remember I told you before that there were two kinds of movements, task-aligned and task-independent. And so now we have a chance to take these uninstructed movements, like the hind limb flexion, see whether they're aligned to the task, like kicking when the lights come on, or whether they're independent, kind of task-independent, more fidgety type of movements. And so the way that we do that, the way that we look at the task-independent contribution is simply to take the task model and to see how much better the task model gets when we add just one single movement parameter. And the intuition there is if there's a movement that is always happening at the same time as a task event, then including that movement in the model isn't really going to improve the performance that much. But in fact, including these movements did help the model fit the data quite a bit. So there's a fairly big task-independent contribution of all of these different kinds of movements, and also pretty big task-dependent. So that tells us that the animal is fidgeting a lot, and is also fidgeting in kind of maybe a superstitious way that it thinks might have something to do with the trial. Looking at the instructed movements, we found, not surprisingly, that they had a big task-aligned component and a smaller task-independent component as well. So going back to this matrix that I showed you at the beginning, this now summarizes all of the variance that's captured by these different classes of movements. And I just want to remind you that even people that thought really deeply about movements and decision-making, that really only focused on that upper left quadrant, which is certainly an important one, but it turns out that there are a whole lot of other movements that are really, really important for driving neural activity that have largely been ignored up until now. Okay, so what does this mean for us? What are the implications? Well, I'm going to tell you about two kinds of implications, and it depends what kind of signal that you're interested in. So suppose you're interested not in average activity, not in the PSTH, but in single trial activity, fluctuations that occur trial to trial. Many people have viewed these in the past as being noise. There's all kinds of hypotheses about trial to trial neural variance and what its function might be. But we now have a pretty different point of view. So we find that these fluctuations are not explained by instructed movements. They're not really explained by the task. These fluctuations are really explained by uninstructed movements. So these fluctuations, which first appear to be noise, are not noise. They reflect the fact that the animal or the human has a whole set of priorities that we don't know about, but that have a big, big impact on what the brain does. Okay, but maybe you don't care because you're all about the PSTH. You only look at trial average activity. You're a little better off, right? Because remember, a lot of these fluctuations, if they're task independent, they're happening at different times every trial. They're going to average out in the PSTH. So you might think you're off the hook. And if you're in V1, that's a little bit true, right? So the model does reassuringly well and predict the task model, reassuringly well in predicting V1 activity. We miss a little blip here, which turns out to be a combination of instructed and uninstructed movements. So that tells us there might be some features of your PSTH that are driven by movements that you didn't know about, but mostly not. But in other parts of the brain, the uninstructed movements matter more. So in M2, for instance, a continuous ramping of activity that occurs over the entire course of the trial probably doesn't reflect a single process that's increasing over time. Instead, it reflects multiple different kinds of processes, all of which are increasing over time, sometimes with rather different temporal dynamics. So I think it's an important reminder that even though you might think you have kind of a uniform drive that's changing your neural activity, it might actually reflect the sum of a couple of rather different drives. So how does experience shape these responses? Well, one really nice feature of doing widefield imaging is that we can do it all the way through learning. So we can see how the brain changes as the animal goes from being a novice decision maker to being an expert decision maker. We can track the contribution of these three different classes of movements over many, 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 many days as the animal's learning a task. And that's what you see here. You can see that uninstructed movements always dominate. although they come to dominate less as time goes by. So as the animal becomes more of an expert, these fidgets tend to matter less. If we compare the activity at early and late, we can see that there are changes all across the brain as the animal's learning the task. And when we look at task-aligned versus task-independent movements, we find, not surprisingly, the task-aligned go up, but interestingly, the task-independent ones go down. So let me be really concrete. So this is what whisking looks like early on. This is the amount of whisking as a function of time now in the trial. And you can see that early on, the animal just kind of randomly whisks. The gray traces are individual trials, and the black trace is the average. But then, when the animal's an expert, this is what whisking looks like. You can see there's a super sharp whisking peak every time the stimulus comes on, and then a little bit more whisking towards the end of the trial. So this task has nothing to do with whisking, right? We didn't tell the animal to whisk. It's a visual and auditory task, but for some reason, making those stereotype movements became part of the task for the animal. And to me, this is reminiscent of a lot of other kinds of human experts who often develop rather extraneous movements as they become experts. Musicians are one expert, too much jet lag, one example of this. But another example is baseball players. Here you see Nomar Garciparra, who's a professional baseball player for the Boston Red Sox. And he's got massive task-aligned, uninstructed movements. Okay, here's another one. So that guy has to hit his feet with the bat before he's able to hit the ball. Those movements have nothing to do with hitting the ball, and yet for those individual subjects, making those movements is a fundamental component of what it means to be engaged in their expert behavior. And so I don't quite know. I think it's an open question, the extent to which this applies to cognitive tasks as well, but I think it's absolutely the case that many of us, when we're engaged in a very thoughtful cognitive task, have a lot of difficulty suppressing movements, right? We click pens and rock chairs and shake legs. A few of you are doing that up there now, I can see you. It's a really natural thing to do, and people find it annoying, so often we feel like we should probably suppress those movements. They're probably bad. Kids definitely get in trouble when they make them in school, but maybe these movements are actually a fundamental part of engaging in that cognitive task in a way that we haven't yet realized. So maybe suppressing them might actually be hurtful to the expert task that we're trying to engage in the moment. Anyway, that part is hypothetical and kind of speculative, but I think it's interesting to think about what purpose these movements are serving, because clearly they're a big part of a lot of tasks, even for experts. Okay, so I've told you so far that movements dominate the wide field signal, but you might wonder, is this only evident because we measure a signal that's pooled over many neurons? What I showed you was not single neuron activity. We have 20 micron resolution, which is pretty good, but there's lots of neurons that are contributing to those signals. We don't really know where they are in terms of depth because we collect signal from all throughout all the layers of the cortex. And you might wonder if this is only true in the cortex, right? We don't get access to subcortical structures with this method, and so just looking at the wide field alone doesn't get at this issue. Okay, so back again to Skeptic's Corner, and I'll address these issues one at a time, starting with this one. So to address whether these results are due to the fact that we were measuring a pooled signal, we broke out our two-photon microscope and repeated all of the experiments that I've told you about, this time looking at single neuron activity. So we identify the neurons, segment the neurons using software written by Marius Pachitariu. We're able to record about 500, 600 single neurons every day. And we did this in lots of different areas. The data that I'm going to tell you about represents over 12,000 neurons that we measured in 5 different areas. ALM is a motor area, MM is a secondary visual area, S1 primary somatosensory cortex, retrosplenial cortex, and of course primary visual cortex. What you see in the traces here, this is just average activity to show you that the neurons in these areas do kind of approximately what you would expect them to do given their known properties. So what we did with the two-photon data is we took exactly the same model that I told you about before, except this time we're not trying to model trial-by-trial fluorescence fluctuations. We're trying to model trial-by-trial neural responses of single individual neurons that we measured with the two-photon. But other than that, it's exactly the same. So I'm going to show you a summary plot for each of the areas that we recorded from. This is the same format that I showed you before. It's the instructed task-instructed and uninstructed movements. On the bottom is the more conservative method, the top is the least conservative. You can see that in area ALM, again, the uninstructed movements dominate. That's also true in the secondary visual area, also true in primary visual cortex, in retrosplenial cortex, and in primary somatosensory cortex. So everywhere we looked with the 2-photon, we still found that you really needed to have the movement signals to understand the neural activity. One really nice feature about measuring neural activity with the 2-photon is that you can focus the microscope to different depths. And this allows you to tackle head-on the question of whether superficial versus deep neurons in deep layers differ in the extent to which they reflect movements. We did not find much of a difference. So here's a summary of all the superficial versus infragranular neurons that we recorded. We can't go too deep with the 2-photon, but we can go pretty deep, so down to about layer 4. And you can see that it doesn't really matter what depth you're at, we still find that the uninstructed movements really dominate the signal. So for the 2-photon, I just want to show you a couple of single cell examples because I think they kind of highlight a really interesting point about how cognitive signals can mask as movement signals and movement signals can mask as cognitive signals. So I'm going to show you now the responses of 3 example neurons and their fits from the 3 different models that I told you about. So here we have one neuron, and here are the fits from the 3 different models. So this neuron was really fit well with the task model. So it isn't the case that every single neuron cares only about movement. Some of them are really strongly task-driven. And if you're someone who's interested in cognitive signals, you can just go and find these neurons and restrict your analyses to those. Here's a second neuron. Interestingly, this neuron has kind of the same trial average time varying firing rate. So it starts off low, kind of ramps up during the stimulus activity, and eventually goes back down. So if you look at just the gray traces in the top row and the bottom row, the gray traces are pretty similar. You might be tempted to put these 2 neurons in kind of the same class of neuron, but they are totally different. While the top neuron is absolutely a task neuron, the neuron below it is completely explained by uninstructed movements. So even though their trial averaged activity is quite similar, they are driven by utterly different aspects of the task. So we need to be really careful when we look at neurons that have time-varying signals like this. Sometimes they have interesting dynamics that resemble the predictions of models that we might like, maybe integration of evidence over time, but we need to be really careful to make sure that we really understand what's driving the neurons. And here's a final example of a cell that's really just a movement kind of burst neuron. It's really driven most strongly by instructed movements. It has a burst at the end of the trial. If you're wondering about cell types, in this particular 2-photon study, we measured the activity of all the neurons together, so all the excitatory neurons. In another paper that's just about to come out, I think next month, we look at the contribution of excitatory and inhibitory neurons separately to decision-making. So if you're interested in kind of testing out some of those classic decision-making models with pools of inhibitory neurons, definitely check that work out as well. Okay, so back to Skeptic's Corner. So I hope I've convinced you that, the movement dominance that we report is not present because we're looking at pooled activity. So next question, is this only true in the cortex? What about deeper regions? So to get at that question, we turn to our NeuroPixels probes, which are a really nice biosensor that has electrodes all the way up and down a long shank. You can record from many, many neurons at a time in the cortex, and then also much deeper as well, because you do multi-area recording whether you want to or not, because the NeuroPixels are long and they have contacts all along the course of the probe. We developed kind of a neat device to hold this probe. We were originally studying them in freely moving ethological experiments, although I won't be telling you about that today. If you're interested in building such a device yourself, that work is also now out, so it has all the instructions for how to do it. So I'll tell you about a couple of animals that we recorded electrophysiological signals from. These are some example days. You can see that we get pretty good, fairly well isolated units, and then we just apply exactly the same model that I've been telling you about all the way through the talk. And again, we find that with the electrophysiological data, subcortical data, again, we find exactly the same thing, that uninstructed movements are really critical for explaining the activity. And this, I think, was a really important control. Calcium signals are a little bit weird. They're quite closely related to spiking, but that relationship is quite complicated. So seeing the same effect when we look at neurophysiological data, electrophysiological data was important. This just shows you one single cell example. This cell's firing rates are fluctuating over the course of the session quite a bit. And if you didn't know about movements, you might offer all sorts of explanations for why that was. But in fact, it's because the amount of uninstructed movements are just changing over the course of the session. Okay, so finally, we wondered, We don't yet know the answer, but a natural question is, are these uninstructed movements, are we seeing such a big signal because this work is done in mice? And we don't know the answer to that. And a natural question is, do humans make uninstructed movements during cognitive tasks? It's an open question. It's one we're hoping to get to the bottom of. I do have a hypothesis. I think humans make a lot of uninstructed movements. I think that this is a collection of movies that I was able to collect over about an 8-minute period, because it turns out that almost everybody fidgets and also wants to talk about it, which is kind of interesting. Maybe because they've gotten a lot of pushback for having their fidgets, but a lot of people fidget, a lot of people move all the time. And I think it's a really intriguing idea that these movements might be more closely connected to cognition than we previously thought. Okay, so I'll summarize what I've told you so far. I've argued that uninstructed movements are critical for interpreting neural activity, that uninstructed movements have both a task-aligned and a task-independent component. And the main message that I hope you'll take home is that during cognition, animals engage in diverse instructed and uninstructed movements with profound consequences for neural activity. If you're interested in reading more about this paper, it's in press at the moment. It's also on the bioarchive. We're also digging much deeper into this, taking advantage of the fact that we can restrict the calcium signal to specific pathways. We have a Society for Neuroscience poster looking at only the activity of cortical neurons projecting to the striatum. So we just look at corticostriatal pathway specifically, which is a really nice follow-up of this technique. So if you're going to SFN, definitely come check out the poster and tell us what you think. These are the people that did the work. The lead person on this team was Simon Musil, who you can see up here, who also worked really closely with Matt Kaufman, who's now a professor at the University of Chicago. They were co-contributors on this project. And thank you very much to all of you, and I'd be happy to take questions.
So do we have the microphone helpers around? Great. So we'll ask a question. I'm wondering, given the new recording modality, can you speak to, let's say, on one extreme, compare this widefield calcium imaging signal to, say, the single spikes, and on the other extreme to, say, fMRI and temporal, spatial, et cetera?
Yeah, so in terms of the spatial and temporal resolution, it's a little bit closer to two-photon imaging, really, because the spatial resolution is about 20 microns. Depends a little bit how you count, but roughly 20 microns. And then the temporal resolution is about 60 hertz. So it's kind of like two-photon imaging, which is also kind of in that range. That's a lot slower than electrophysiology. If you care about really fast temporal dynamics, those are going to be masked, mostly because the calcium signal itself is kind of slow. It's a lot faster and more spatially precise than fMRI, but I think really the big win here, I mean, fMRI is an amazing technique, and with fMRI you can look at humans, and surely looking at humans is more relevant to the human brain than looking at the mouse brain. So, but I think there's one really big win, which is that we can take advantage of the genetic specificity by using the tools to restrict the indicators to particular pathways. So I think what I told you about today is really a first step and kind of has highlighted the importance of the movements and opened up kind of a new world of using video, but I think the real power of this technique is yet to come. I'm going to leave you in charge of questions. Oh, can I say one thing, though? I would really like to encourage trainees to ask questions. I find trainees often have a great perspective, so if you're a trainee, it'd be great to hear from you, too. And also from you. If you're not a trainee, I don't know.
Great. I'm not. Great talk. So you said that movements dominate the neural activity and that the movements may be important for executing cognitive tasks. And I would like to push you on both those points, because isn't it possible that these are actually sensory signals driven by those involuntary movements? And of course, the sensory signals will be correlated with the movement signals. And the other thing is that maybe they are correlated with the task, but not driving the task. So, for example, there may just be a lot of activity going on in the brain, and rather than trying to suppress the activity, so let's say it's all just junk activity, but rather than suppressing it, our brain just lets them happen because it's not worth spending the energy to suppress the activity.
Yeah, so let me answer those one at a time. So I think, first of all, your question about whether the signals that we see are driving the movements or are sensory consequences of the movements, they're definitely both. Oh, wait, hang on, that's actually not the slide I wanted, just a sec. And you can totally tell that by looking at the temporal filters. Okay, so here's an example. Hopefully you guys can see that. Okay, so this is the temporal filter you see here is related to whisking. So we looked, we used the model to fit the filters in the same way that we always do. And what I've shown at the bottom is the filter corresponding to two different parts of the brain, to the retrosplenial cortex and to the barrel cortex. And you can see that the temporal filters just have really different dynamics. So with retrosplenial cortex, it's tending to peak a little bit before the movement. With barrel cortex, it's tending to peak a little bit after the movement. With other kinds of movements that we look at, it just depends what part of the brain you're looking at. But we can get a pretty good handle on that. We don't have the precision that we would really like to get because we don't have spikes, we have a calcium signal, but we can certainly begin to distinguish preparatory versus sensory consequences of the movement. In terms of the usefulness of these movements, I mean, I completely agree. I think the point that we're making is really speculative. The idea that maybe these movements are either a consequence of cognition or aiding cognition in some way is a hypothesis that we find intriguing, but we don't yet have evidence for. But I think one observation that supports that point of view is that movements are very costly. So in general, our motor system is highly, highly optimized. Like when we walk upstairs, we only pick up our foot just enough to clear the stair, such that if you change the stair height, everybody falls down. So we're like super optimized. We're like not wasting any energy. We reach for things in the most energy efficient way. And yet here we are like rocking our legs all the time. Like these movements come at a big metabolic cost. And in fact, it's even known that people who are high fidgeters tend to use up a lot more energy. In the modern world, of course, we have a lot of food. There's not a huge consequence of that. But it's clear that they come at a big metabolic cost. And so my hypothesis is that if these movements are so costly, they must have some function. The brain must have a reason for executing them.
Okay, so let's go with Anne's suggestion for a question from a trainee.
So what do you think would happen if you were able to paralyze the entire animal except for the tongue, and both behaviorally and neurally?
Oh, I don't know. That's an interesting question. I mean, I think in that case, we would see an efference copy of the signals that were executed. So for certain aspects of the filter, we might still see some activity there. For other kinds of movements where the activity reflects the sensory consequence of the movement, probably we would see a lot less activity.
Any more questions?
Do you think they'd suffer behaviorally?
Oh, I see what you're asking. Okay, so if they were allowed to do the instructed movements, but were somehow forbidden, yeah. Okay, no, that's an interesting question. I think that is my hypothesis, yeah, that these movements are supporting them. And so if you were able to suppress those movements in some way, my hypothesis is that the behavior would suffer, yeah.
Thanks for a great talk. And I have a question regarding your regression model. So I'm wondering, so do you mind commenting on why is it a fair comparison for the contributions for the directed for the instructed and uninstructed activities? Because some might argue that the difference might be attributed to the different feature engineering of the model. So for example, like the video is a two-dimensional space, and also all the noise and whiskering, they are in continuous space, but say the licking and handles, they might be binary labels. So the regressors might be tricked to be attributing more weights on those with more continuous space.
Yeah, it's a really important question, and that's one that we thought about a lot. In fact, the temporal filters, the linear temporal filters, they really are the most flexible because they can account for peaks of activity that occur at any moment in time. So the analog predictors, they are fluctuating continuously, but the only thing you can really do to them is scale them. So like if the pupil and activity somewhere are going up and down together, then you're totally set. But if there's any kind of time delay, then the filterless analog predictors are totally useless. But we did do a control where we only included the uninstructed movements that were of the temporal kernel sort, and there's quite a lot of those, right? Like you can see, in fact, in the example with the whisking here, for The whisk in this analysis, the whisking is binarized, so that even the whisking, I mean, you can see there, you can use it both as a continuous and as a binary vector. And we see a big contribution from that uninstructed movement, whether we include it in the filterless analog category or the linear temporal filter category. So I don't think it's the difference in the class of regressors that explains the result.
I think we have time for one more question.
I have a question about your differences in performance in the task, because you mentioned at the beginning that there is something like action slips. So can you relate, for example, the spontaneous movements to performance in differences in the animals?
Sorry, can you ask the question one more time? I didn't quite follow that.
So can we predict based on, for example, uninstructed movements if animals perform the task well? So are they more like helping people with the task or harming them?
Yeah, so there's a great paper on that very topic from Fritjof Helmchen's lab, and the first author is Ariel Gilad. And in that paper, they grouped animals into high movement animals and low movement animals. And that the main take home from that study was that those two groups used different aspects of the brain to support the task, which is itself very interesting. But they also did look at, among those two groups, which were the higher performers, and also within each group, were they doing better or worse when they were in their respective categories. So first of all, the high movement group did a tiny bit better. One notable aspect of this is that within the high movement group, when that group was not moving, so when they were still, they tended to do worse relative to when they were moving. So I think that's a piece of evidence that if you happen to be a high fidgeter, you should probably keep fidgeting. But I do take it with, so everybody can like click their pens now. But I do take that with a grain of salt, because the problem is with a high fidgeting individual, maybe the reason that you're now still is just because you've checked out. So it's hard to distinguish those two possibilities. But I think that study begins to suggest that there might be some benefits to moving around, at least if you're in that movement type of group.
Okay, so we are nominally up for a 10-minute break, and then the poster sessions begin. So let's thank Dr. Churchland one more time.